My topic of discussion is life cycle strategic plan of nilotinib or Tasigna. Will be discussing early phase and preclinical phase, and in these slides I'm going to discuss early development phase of nilotinib. Summary of my discussion will include phase 1 study and phase 2 study. Within phase 1 study, I will be discussing participant selection criteria, study design and dose selection study, pharmacokinetic and pharmacodynamic studies, including toxicological assessments, results. Within phase 2 study, I will be discussing phase 2 study that were conducted for 3 different disease states, which is chronic, accelerated, and blastic. Let's begin with phase 1 study participant selection criteria. There were few criteria set to determine whether a patient should be included or excluded from the study. In addition, since this drug is prescribed in cancer disease, phase 1 study was conducted in patients rather than healthy volunteers. So first criteria was to have Philadelphia chromosome positive gene. Second one was to have imatinib resistant patient. Imatinib resistant was defined as lack of complete hematological response after 3 months of treatment, or lack of any cytogenetic response after 6 months of treatment. Patients who were in blastic or accelerated phase of the disease were also included. Patient must be at least 18 years of age and have normal hepatic, renal, and cardiac functions. Patients who had received imatinib treatment or hydroxyurea treatment were excluded from the study, since these drugs may interfere with function of nilotinib and it may interfere with the result of the study. Next is the study design of phase 1 study. Total 119 patients were enrolled in phase 1 study based on the selection criteria described earlier, and each patient was given a dose of nilotinib as shown in the figure. Patient received a daily dose of nilotinib ranging from 50 mg to 1200 mg, or twice daily doses of 400 mg and 600 mg. Per the diagram, 7 patients received 50 mg dose, another 7 patients received 100 mg dose, and then another 10 patients received 200 mg dose, and so on and so forth as described in the figure. Patients who received lower doses were allowed to take higher doses after appropriate wash period is achieved. In addition, patients were also allowed to take cytoreductive therapy if an elevated count of blasts and/or platelets were found in their blood during the study. Dose selection study. Patients received 50 to 1200 mg daily doses through an oral route. However, doses greater than 400 mg showed similar AUC level due to serum saturation, as shown in figure number 1. Therefore, twice daily doses were analyzed, and twice daily doses produced 50 to 80% improvement in AUC level and produced better dose response relationship. Therefore, twice daily doses were selected. Among the two doses of 400 and 600 milligrams, 400 milligram dose had a better safety profile and compared to 600 milligram dose. Thus, 400 milligram was selected as a preferred dose for phase 2 trials. As you can see in the table below, 400 milligram produced relatively less side effects or side effects in less population compared to 600 milligram dose. Next is assessment and pharmacokinetics. Patients who received nilotinib daily or twice daily doses, their blood samples and biochemical assays were collected for first 8 weeks and then every other week. Their bone marrow were also collected on day 15 and day 28, and then on day 28 for every even numbered cycles. Safety assessments included adverse event evaluation, hematological and cardiac enzyme assessment, ECG, urine analysis, physical examination, and biochemical testing. Based on the samples collected, following pharmacokinetic parameters were calculated. The peak concentration was reached after 3 hours of dose administration. At steady state, peak concentration was 3.6 micromolar in patients who received 400 milligram twice daily doses. Half-life of the drug was 15 hours and steady state level reached by day 8. Next is pharmacodynamics. 400 milligram of twice daily oral dose was well tolerated in most of the population. Uh, Nilotinib is absorbed is given orally and it is absorbed through GI tracts and have a higher bioavailability when taken after 30 minutes of high-fat meal compared to the fasted state. From in vitro study, it was proven that serum protein binding is known to be 95%, and nilotinib is metabolized in liver through oxidation and hydroxylation. None of the metabolites were considered active or had any activity. Most of the drug is excreted in feces within 7 days of the dose administration. Next is the safety. After administration of nilotinib, number of side effects were reported and observed during phase 1 study. Some of them are thrombocytopenia, neutropenia, rashes, muscle spasm, GI disturbances, fatigue, increase in indirect bilirubin. Nilotinib also prolonged QT and F interval in some patients, and death of 2 patients was reported during this trial. As it can be seen in the table below, based on the dose, side effect increases, and therefore 400 milligram twice daily dose was selected since it had lowest side effects and highest drug efficacy. Phase 1 study results. As stated earlier, 2 patient populations were selected, patients having accelerated and blastic phase of the disease. Accelerated phase patients showed 74% hematological response and 55% patients showed cytogenetic response, whereas in blastic phase 39% patients showed hematological response and 27% patients showed cytogenetic response. Phase 2 study. As described earlier, phase 2 study, there were 3 phase 2 studies conducted based on the disease states such as chronic, accelerated, and blastic. Chronic phase, total 321 patients were enrolled having chronic phase of chronic myeloid leukemia. Dose of 400 and 600 milligram twice daily were given to the patients, and median rate for complete hematological response and complete cytogenetic response rates were 1 month and 2.8 months respectively. The estimated 18 months overall survival rate was 91%. In accelerated phase, 136 patients were enrolled and 400 milligram twice daily dose was given. Median time for complete hematological response and complete cytogenetic response rate were 1 month and 2.8 months again, and the estimated 12-month progression-free and overall survival rate was 81%. Phase 2 study, Blastic Phase, 136 patients were enrolled in total and they were given with 400 milligram twice daily doses. The estimated 12 the estimated 12-month survival rate was 42%.